Let's see how different proteins have different structures. Each protein has a specific protein properties which are determined by the number and the specific sequence of amino acid in a molecule and upon the shape which the molecule assumes as the chain forms into its final compact form. There are four levels of organization which are described below. Primary structure. The primary structure comprises the number and sequence of amino acids in a protein molecule. F. Sanger was the first scientist who determined the sequence of amino acids in a protein molecule. After 10 years of careful work, he concluded that the insulin is composed of 51 amino acids in two chains. One of the chains had 21 amino acids and the other had 30 amino acids, and they were held together by disulfide bridges. Hemoglobin is composed of four chains, two alpha and two beta chains. Each alpha chain contains 141 amino acids, while each beta chain contains 146 amino acids. The size of protein molecule is determined by the type of amino acids and the number of amino acids comprising that particular protein molecule. Now we know that there are over 10,000 proteins in the human body, which are composed of unique and specific arrangement of 20 types of amino acids. The sequence is determined by the order of nucleotides in the DNA. The arrangement of amino acids in a protein molecule is highly specific for its proper functioning. If any amino acid is not in its normal place, the protein fails to carry on its normal function. Best example is the sickle cell hemoglobin of human beings. In this case, only one amino acid in each beta chain out of 574 amino acids do not occupy the normal place in the protein. In fact, this particular amino acid is replaced by some other acid and the hemoglobin fails to carry any or sufficient oxygen, hence leading to the death of the patient. Secondary structure. The polypeptide chains in a protein molecule usually do not lie flat. They usually coil into a helix or into some other regular configuration. One of the common secondary structure is the alpha helix. It involves the spiral formation of the basic polypeptide chain. The alpha helix is a very unique geometry structure with 3.6 amino acids in each turn of the helix. The helical structure is kept by the formation of hydrogen bonds among amino acid molecules in successive turns of spiral. Beta pleated sheet is formed by folding back of polypeptide. Tertiary structure. Usually a polypeptide chain bends and folds upon itself, forming a globular shape. This is the protein's tertiary conformation. It is maintained by three types of bond, namely ionic, hydrogen, and disulfide. For example, in aqueous environment, the most stable tertiary conformation is that in which hydrophobic amino acids are buried inside while the hydrophilic amino acids are on the surface of the molecule. Quaternary structure. In mainly complex proteins, polypeptide tertiary chains are aggregated and held together by hydrophobic interactions, hydrogen and ionic bonds. This specific arrangement is the quaternary structure. Hemoglobin, the oxygen-carrying protein of red blood cell, exhibits this structure.